Hey everyone, welcome to Biology 1 lecture. So each week I'll be recording a couple, 2 or 3 video lectures typically. I try to make them short and then you spend more time working on questions, um, which is what we normally try to do in class, but obviously this semester we're doing that online. So we start off the semester with a couple chapters which are mainly backgrounds of chemistry. So this is kind of just to remind us of chemistry, and chemistry is really the basis for a lot of what we're talking about this semester. So a few definitions to start. An element, a substance that can't be broken down to other substances by chemical reactions. So the elements that we think of in biology a lot are carbon, hydrogen, oxygen, nitrogen, etc. An atom is the smallest unit of that element. So it's the smallest unit of matter that retains the properties of an element. It contains protons, neutrons, and electrons. Molecule are when you take 2 atoms and you combine them through a covalent bond. So molecules are made up of covalent bonds between more than one atom. And compounds are when you combine 2 or more different elements into a fixed ratio, either by covalent bonds or by ionic bonds. So the chemistry in biology is actually quite a bit of a much smaller subset of all chemistry. So there's, of the 92 elements found, and that number is actually probably lower than in reality now, only 25 are known to be essential for life. 4 elements make up 96% of living matter. So which 4 elements are those? Everyone holler them out. Well, I can't hear you, but I'm sure everyone got them right. So the 4 most important ones or the most common ones are carbon, oxygen, hydrogen, and nitrogen. And you can see over here, these are the 11 elements that make up all but 0.01% of the human body. So these are really what we're talking about when we're talking about the chemistry of biology. So again, elements are substances that can't be broken down further by chemical reactions. Compounds are when 2 or more different elements are combined in a fixed ratio. And so they're combined through chemical reactions. And compounds can be changed, and their properties can be changed by chemical reactions. So here's a simple chemical reaction where sodium and chloride are changed into sodium ions and chloride ions, which can then bind together through ionic bonds. And so sodium is a metal, chloride is a gas, and sodium chloride is actually table salt. So, we're putting this on our steaks or salads, and these are quite different. So chemical reactions change elements. So atom is the smallest unit of matter that still retains the property of an element. So atom's that fundamental unit of an element. Molecules are when you take 2 atoms and link them together by a covalent bond. So 2 hydrogen atoms here. Here we have a carbon and 4 hydrogen atoms. So these are molecules which are linked by covalent bonds. Atoms are made up of protons, neutrons, and electrons. And so here is a simplified diagram of a helium atom. Helium contains 2 protons, the positively charged particles in the nucleus. It also contains 2 neutrons, the neutral particles within the nucleus, and then it contains 2 electrons, which are zipping around outside, shown here in an energy diagram, but they really are somewhere out here in space. Atoms are mostly space, so if this helium atom was the size of a superdome, the nucleus, where almost all of the mass is about the size of a pencil eraser, and the electrons are little gnats that are flying around in all the empty space of the superdome. So atoms are mostly space, and almost all of the mass is within the nucleus. So there's some terms that we use to define atoms. And so these 3 things are important to remember because otherwise it gets confusing when you're doing problems, etc. So atomic number is the number of protons And so the number of protons, if it's a neutral ion or neutral atom, is going to be also equal to the number of electrons. Protons are positively charged, electrons are negatively charged, and so if the atom is neutral, these two are equal. The mass number is the number of protons plus the number of neutrons. And then the atomic mass is the mass of the atom, which is essentially the mass of the protons plus the neutrons. So atomic number, again, is the number of protons, which also for a neutral atom equals the number of electrons. It's specific for that element. So the atomic number of carbon is always 6. Carbon always has 6 protons. That's what defines that atom as being a carbon atom. Isotopes of carbon will differ in the number of neutrons. So for example, for carbon-12, which is the most common naturally occurring isotope of carbon, it has 6 protons. Again, the atomic number is always 6, and it has 6 neutrons. There's another isotope of carbon, carbon-14, which has 6 protons and 8 neutrons. So again, 6 plus 8 is 14, so carbon-14 is an isotope. Carbon-12 is the most commonly found. Carbon-14 turns out is radioactive with a really slow half-life, and so it's used in a lot of carbon dating of things. Okay, so again, the atomic number of protons determines the which atom that is. The atomic mass is the actual mass of all the protons and neutrons in the atomic nucleus. So electrons are quite small, 1/2000th the mass of a proton or a neutron. So the mass of the atom is essentially equal to the mass of protons plus neutrons. In biology, we talk about atomic mass in units of daltons. In chemistry, they'll talk about atomic mass units. Those are essentially the same thing with different names. And so 1 dalton is equal to the mass of 1 proton or 1 neutron. Protons and neutrons are about the same mass. And in biology, we refer to those as 1 dalton. And so it turns out the atomic mass, which is the mass of protons plus neutrons, is very close to the mass number, which is just the number of protons plus neutrons. So those two numbers are almost the same because protons are 1 dalton and neutrons are 1 dalton. When you look at a periodic table, sometimes you'll see slightly different atomic masses. So carbon is 12.0 something. And that's because they're averaging the different isotopes. So there's mostly carbon-12 found naturally occurring, but there's a little bit of carbon-14, so it's a little bit different than just 12 itself. The electrons of an atom can be at different energy levels. So energy is defined as the capacity to cause change or do work. Potential energy, and this is in particular for what we're talking about, is the energy that mass possesses because of its location or structure. So you can think of water behind a dam of having potential energy. The ball on the top of the steps has a lot of potential energy. In order to get that potential energy, someone had to move it up. So work had to be done to get the ball to the top step. And now if the ball goes down or water goes down a dam, that energy can be used to do work. And so electrons are in distinct energy levels. Those energy levels are often described as different electron shells, kind of like how this would be the high electron shell, the medium electron shell, the low electron shell is with regard to energy. And so you can think of electron shells as analogous to this ball on the steps. So here we normally draw electron shells in circles surrounding the nucleus. An atom can move from one shell to another if it gains or if it loses exactly the amount of energy that's different just between those two shells. So the first step of the ball's on the first step, the ball's on the second step, the ball's on the third step. Again, these shells are just ways that we can represent energy levels. Electrons aren't really zipping around in a perfect orbital like this. The geometry is complex and it's determined by probabilities more than distinct patterns of movement. So electrons are more stable at the the lower energy shells, which are closer to the nucleus. And so the positive charge is stabilizing with the negative charge there. As you move away from that positive charge, you get to the higher energy levels. The periodic table here, a portion of the periodic table shown here, is built such that it shows the electron distribution for the elements. So that's the key thing. The number of electrons and at what energy level they are determines the reactivity of that particular element. And so the periodic table represents the elements in that way. And so in these diagrams within this version of the periodic table, each energy level is defined by this circle. So in the first level, we have first row, we have 1 energy level. Second row, we have 2 energy levels. Third row, we have 3 energy levels. So first energy level is the lowest energy level. Electrons in the second energy level are of higher energy than those in the first, and electrons in the third energy level are higher again. So the first row of elements has only 1 energy level, and that first energy level can only have 2 electrons max. So hydrogen has 1, helium has 2. So that energy level only has a room for 2 electrons total. The second row, the second energy level, has room for 8 electrons, and so you can see the electrons drawn in here as the little dots. You can fill up the second with 8 little dots, and the third energy level can again take 8. And so the first energy level takes 2 electrons, second 8 electrons, third 8 electrons. And that's important for determining what the valence is, which is the number of electrons in the outermost shell. So let's talk about that. Valence electrons are electrons in the outermost shell, which is also called the valence shell. It's the highest energy, and so the valence electrons for a particular element are the highest energy electrons. And those are going to determine the chemical behavior of an atom. So here for hydrogen, its valence electron is only in the first energy level. Here for lithium, its valence electron is in the second energy level. Here for sodium, its valence electron is in the third energy level. So they differ by where their electrons are, or where their valence is first, second, third. So to go through a couple. So the atomic number for hydrogen is 1, so it has 1 proton, which means it also has 1 electron. The first shell is its valence shell. Now the first shell can take 2 electrons, hydrogen only has 1, and so it really wants to have 2 electrons. Atoms are trying to reach their full valence shell to be the most stable, and so for hydrogen it can become full in its valence by gaining electron from another atom and sharing that electron. And so if hydrogen was to share electrons with another atom, it would form a bond. What type of bond is that? Does anyone know? Everyone holler it out. Yes, indeed, covalent bond. And so that's the driving force for forming covalent bonds, is it's a way for valences to become full and for that atom to become more stable by forming a molecule with another atom. Helium has an atomic number of 2, so 2 protons, meaning it has 2 electrons. The first energy level is its valence. It has 2 electrons, so that's at its max. And so helium is non-reactive. It's actually a part of the noble gases, which are non-reactive gases, because it's already happy. It's got all the electrons it needs. Nitrogen, atomic number of 7, so it has 7 protons, also 7 electrons. Its first shell has 2 electrons, shown here. Its second shell, so if we have 7 electrons total, 2 in the first, that leaves 5 for the second. So the second we have 1, 2, 3, 4, 5. So it has 5, and it needs to have 8 in that shell. So if it wants to be happy, it needs to gain 3 electrons by forming covalent bonds and sharing 3 electrons with other atoms. Sulfur, down here in the third row, atomic number of 16, so it has 16 protons, thus 16 electrons. First shell has 2 electrons, second shell has 8, so 2 and 8, that's 10. I have 16 total, so it leaves a 6 in the third shell. So the third shell is its valence. It has 6 electrons, so it needs to gain 2 to reach its 8 to be full. And so sulfur is looking to gain 2 electrons by forming covalent bonds. So again, the periodic table tells us about reactivity. So the ones in this final column over here, the noble gases, they have all their valences are full. So the first one's full here for helium, second one's full here for neon, third one's full here for argon. So these are basically non-reactive. Here is examples of elements that need to gain 1, 2, 3, or 4 to fill up their valence. So these guys are all looking to share electrons, form covalent bonds in order to satisfy their valence shell. So some of the problems in the video lecture quiz are going to be, trying to draw these diagrams and determine what you predict about sharing of electrons, what atoms are going to form covalent bonds with which other atoms. So it'll be good practice to think about this idea of valence electrons and atomic mass and atomic number. Okay, so as we just said, atoms are most stable if their valence is full. They can satisfy their valence by sharing or transferring electrons with another atom via a chemical bond. So sharing of electrons forms covalent bonds. Transferring of electrons makes ions. So if you take an electron away, that's going to leave that atom positively charged because the number of protons remains the same. So ions are ones that have a +1 or -1 or +2 or -2. And so ions, once they form because they've gained or lost an electron, they can then form ionic bonds with other ions. So the negative charge and the positive charge can interact electrostatically and form bonds. But in both of these ways, you're achieving a full valence. So we already kind of went over covalent bonds, but covalent bonds, again, are the sharing of a pair of electrons. So here's 2 hydrogens. Each one has 1 electron. If they come close together and share those 2 electrons, so the electron that used to be here can now buzz around here. The electron that used to be here can also buzz around here. So those 2 electrons are being shared by these 2 atoms, and that's forming a covalent bond. Single covalent bond is the sharing of 1 pair of electrons. A double covalent bond is the sharing of 2 pair of electrons. So oxygen has a valence of 6, so it needs to gain 2 to have its outer shell full. So it shares 2 electrons here with another oxygen, forming a double covalent bond. And so when you see these structural formulas where the covalent bond is represented by a line, each line refers to 2 electrons. So this is 2 electrons being shared, and this is 4 electrons being shared. So there are chemical bonds that are key to how biology works. We've already talked about covalent bonds, so what are the other important chemical bonds for biology? Everyone yell them out. Damn, I still can't hear you. Okay, so we talked about ionic bonds. So ionic bonds are very important. A third type are hydrogen bonds, which are weaker than either covalent or ionic bonds. And then a fourth type are van der Waals interactions, which are weaker still. And so these are the main 4 bonds. 2 of them are actually relatively weak, but they are really important for how biological molecules interact and work in biological systems. So we talked about covalent bonds, ionic bonds. Ionic bonds come from the creation of ions. And so here we have a sodium ion that has 1 electron in its outer shell, the third shell. And so it has 1 electron. It can either gain 7 electrons to be full, or if it loses 1, it's automatically full because then it's full in its second. Chloride is in the opposite scenario where it has 7 electrons in its outer shell, so it just needs to gain 1. So a convenient arrangement is for sodium to give up that electron because that makes it much more stable, and for chloride to pick up that electron, which makes it much more stable. And so when that occurs, because sodium has lost an electron, now it has 1 extra positive charge in its nucleus, and so it's +1 charge. Chloride is the opposite. It's gained an electron, so it has one more electron than it does protons in its nucleus, and so it's negatively charged. Ions that are created by the complete transfer can then interact with each other. And so ions are atoms with more or fewer electrons than the number of protons. Anions are negatively charged ions like chloride that have picked up an electron. Cations are positively charged like sodium, which have lost an electron. And so the ionic bond is formed by the interaction between this positive charged ion and this negatively charged ion. However, it doesn't mean that the two ions that are interacting necessarily were the two that exchanged electrons. The ionic bond is really just the formation, the electrostatic attraction between positively charged and negatively charged ions. Those ions are created by this complete transfer. So the next type of bond that's important is a hydrogen bond. In order to understand what a hydrogen bond is, we first have to think about electronegativity of atoms and how they form polar covalent bonds. So electronegativity is an inherent property of an atom. It usually can be measured. And so it's the attraction of a particular kind of atom for the electrons within a covalent bond. And so an atom that is more electronegative will be better at attracting that shared electron than an atom that is less electronegative. So an example of that is shown here in this water molecule, where here we have an oxygen, which is a very electronegative atom, and that's formed two covalent bonds with hydrogens, which are not so electronegative. And so because this oxygen is really good at sort of stealing away the electrons that's shared, it's partially negatively charged because it's got the electrons more often than the hydrogens, and for the same reason, the hydrogens are partially positively charged. So these covalent bonds shown here with the blue arrows are polar, in that this end of it at the hydrogen is positive, this end of it at the oxygen are negative. So that's a polar covalent bond. Nonpolar covalent bonds form between two atoms that have similar electronegativities. So it turns out that carbon and hydrogen have similar electronegativities. So in a molecule that just has carbons and hydrogens forming covalent bonds, each of these covalent bonds is nonpolar, because neither one of these two are better at getting the electrons than the other. So there's no polarity along the axis of these bonds. And so it turns out that molecules made of just carbons and hydrogens are very nonpolar, and we'll talk about that in our next chapter. So what is a hydrogen bond? So a hydrogen bond is a relatively weak bond that forms between two molecules. So it's not within a molecule, it's between two molecules, between like a water molecule and an ammonia molecule. So there's an attraction between these two that can occur because this hydrogen is partially positively charged, this nitrogen is partially negatively charged. So that interaction between the partial positive and the partial negative, that is a hydrogen bond. Partial positive is almost always hydrogen in biological systems. The partial negative is typically oxygen or nitrogen. And so we typically have hydrogen bonds forming between a hydrogen of one molecule and either a nitrogen or an oxygen of another molecule. But remember, hydrogen bonds are always between two molecules. It's not within. This bond here is a covalent bond. And so here is the hydrogen bond between the hydrogen of water and the nitrogen of ammonia. So as we just saw, water is polar. All the hydrogens in the white cartoon here are all partially positive. All the oxygens, the red, are partially negative. And so water actually likes to form hydrogen bonds with itself. Here, this hydrogen is interacting with this oxygen. This hydrogen is interacting with this oxygen. This oxygen is interacting with that hydrogen. So water is forming covalent bonds with itself, and water has some special properties because of that, of how it interacts with itself. So the reason we have a meniscus and things like that are because water likes to interact with itself. Water is also a good solvent for that same reason. So biological systems are aqueous systems. So the solvent inside of our cells, in our bloodstream, everywhere is water. So the whole system is basically made up of water. And so water consists of all these water molecules, these red and white molecules. So there's huge amounts of water in an aqueous environment. And because waters are positive on the hydrogens, negative on the oxygens, it can interact with negatively charged things. So all these hydrogens are interacting with this chloride ion. All these oxygens, which are negative, are interacting with this positively charged sodium ion. So even though that the ionic interactions between sodium and chloride is stronger than a typical hydrogen bond, when you put that crystal of table salt in water, there's way more waters, and they're going to outcompete the sodiums and chlorides and sort of pull these apart. So that little salt crystal is going to be pulled apart because water is interacting with the sodiums and the chlorides via that polar covalent bond interaction. Water can also do the same thing for large macromolecules like protein. So here's an enzyme called lysozyme, and it's an enzyme that works in the cytoplasm, and it can interact with all the waters to making it a soluble enzyme. So water is a good solvent for large molecules like proteins. And you can see the water is going to interact with, partially negative parts of the protein with the hydrogen of the water, and partially positive parts of the protein with the oxygen of the water. The last bond are van der Waals interactions, and those are very weak interactions from two molecules that come very close together. So it's based on the fact that atoms are kind of partially positive and negative, sort of depending on where those electrons are. And so you can have these partial regions of polarity within an atom interact with another atom. But that interaction only occurs when they're very, very close. So you have to be very close to each other. They're extremely weak interactions, but they become important in biology because biology is made up of these big, giant macromolecules. And so when you have millions of these very weak interactions, they can turn out to be strong. So van der Waals are things that basically two surfaces have to be very complementary to each other, and then they can get that interaction because they're close enough. And they can be quite strong if you add up enough of them. So geckos that you can see on your front porch walking upside down, those stick to that roof of your porch through those van der Waals interactions. So there's lots of weak chemical bonds that are important in biology. They're particularly important for large molecules because all those weak interactions can add up. It also reinforces the shapes of large molecules. So large molecules kind of fold up and form shapes by interacting with themselves, and it's a lot of these weak interactions that occurs. Also important for when two big molecules bind to each other. So interactions between molecules, those weak ones are important. Examples of interactions are the base pairing between the two ladders of the DNA helix, A's with T's and C's with G's. Those are based on hydrogen bonds, which are fairly weak, but you have so many nucleotides along the length of a chromosome that it becomes very strong. Hydrogen bonds and van der Waals interactions are really important for how proteins form their shape, and we'll talk a lot about that in the next section. Sort of an underlying theme for the whole course is the idea that structure determines function. So molecular shape determines the function of that molecule. And the reason is that all biological functions, when you get down to the molecular level, work by one molecule binding to another in a very similar interaction as a lock and a key. And so the shapes of those molecules that are interacting is totally important. So the shape of the lock and the shape of the key have to be exact in order for those two things to come together. And so if function is determined by that binding interaction, the shapes of the two things that are binding is totally critical. So structure and function is really important for molecular biology and cell biology, and we'll hear again and again about that. Structure determines function. And then the last type of interaction is not strictly a bond. It's more of a property and interactions based on how molecules interact with the water. So the idea of hydrophilic and hydrophobic substances. So a hydrophilic substance is polar, so it has an affinity for water, sodium and chloride here, or polar molecules, water likes to interact with them. Hydrophobic substances are things that have nonpolar bonds. So here, all carbons and hydrogens. These are nonpolar covalent bonds. Water doesn't like to interact with them and would prefer to interact with itself. And so because water wants to interact with itself and doesn't want to interact with these dudes, water is going to form a shell around that and sort of squeeze out the nonpolar things. So the reason that oil floats to the top is because water is kind of squeezing out the oil, or you see oil in little droplets, whereas other things that are polar can interact with the water. And so there's some components within biological systems, for instance, cell membranes, which are highly hydrophobic lipids. And so the cell membrane is a hydrophobic thing that doesn't like to interact with water. And so why do you think that would be an advantageous thing for a cell membrane? Why would it be good that water doesn't interact with that? And you are correct. The reason is because we have two aqueous environments, one inside of the cell, one outside of the cell. The cell membrane separates those two, so then you can specialize the chemistry that goes inside of your cell because they're not mixing the two aqueous environments. And so that barrier separating the chemistry of the inside of the cell was really important to how cells evolved and is important to how cells function. So that's it for this video lecture, and there'll be some video lecture quiz questions to go over this material and practice a little bit about what we've talked about. Thanks a lot.